The other major group of cells that make up the antigen-specific immune response are B cells. So B cells are going to be part of what we were calling the humoral immune response. B cells will differentiate into plasma cells, and plasma cells are just basically giant antibody-producing factories. So antibodies are also sometimes called immunoglobulins. They're a type of protein. They have numerous different functions that are listed here in this box. One of the things that they do is opsonization. And opsonization is the idea that they'll coat whatever it is that's invading, and that triggers phagocytosis. So it promotes phagocytosis. They can also neutralize. Neutralization is really important when we're talking about a response to toxins or viruses. In general, we don't see a lot of phagocytosis of toxins or viruses, but we do see neutralization. So an antibody will bind to one of those things, and that prevents the virus or the toxin, sometimes even the bacteria, to our cells. They can agglutinate bacteria, basically cause clumps to form. And when you get clumps, it's much easier for the phagocytic cells to recognize and get rid of them. They can do things like bind to the flagellum, which will prevent an organism that used to be motile. Now it makes it non-motile. We talked about the complement cascade. So binding of an antibody to particularly a bacterial cell and activating complement is the classical pathway. And we can also see antibodies that will combine with antigens on the microbial surface, leading to not only that membrane attack complex, but also recruitment of more phagocytes and more antibodies to the site. So antibodies are really, really important for our immune system to function. We have to have that presence of both B and T cells to have the best type of response. In mammals, particularly humans, there are 5 major categories or classes of antibodies. There's IgA, and there are 2 or 3 subgroups of some of these. So there's IgA, I'm going in alphabetical order, IgD, IgE, IgG, and IgM. Each of these types of antibodies plays a different role in the immune system. They're used at different times during an immune response. For example, IgE is really important for parasitic responses, whereas IgA is secretory. And so what you can think about when we start to look at properties and functions is, what are the consequences of lacking particular antibodies? And we do see that there are patients who can have immunodeficiencies where they're only deficient in something like IgA. So, what happens if a patient is deficient in IgA? What would that make them more susceptible to? So when you think about an antibody structure, we typically draw it, I'll go back a couple slides, you see it generally drawn as this Y-type structure. And that bottom part of the Y is called the Fc receptor. It's the constant region. And you can see here what the functions of some of those are. And, importantly, you see things like mast cells recognize the IgE region. So that's supposed to be super antiparasitic. It also is what can lead to a lot of our allergic responses. In IgG, that can transit from mother to the neonate and protect the neonate. And then, of course, you get complement activation when the Fc region is recognized by things like macrophages or other types of cells. Here again, you see now the whole immune system, including the antigen presenting cells, which are part of that innate immune response, various cytokines, the helper T cells, activating perhaps cytotoxic T cells, activating B cells to differentiate and produce various antibodies, and how the antibodies respond and what we see happen. So you have an antigen presenting cell. If we're presenting on MHC class 1, we might present to a cytotoxic T cell directly. If we're on class 2, we'll present to our helper T cells. Helper T cells can continue to propagate themselves. They can activate more antigen presenting cells. They can activate B cells, activate CD8 positive T cells, mast cells, eosinophils, more macrophages. And so you can see that there's this, again, constant cycle of activation depending on the type of infection we're dealing with. And just like we see with the T cells, and it takes time for a T cell response to occur to a very particular antigen, we see that delay in the antibody response as well. So the first time an antigen is encountered, let's say at day 0, it can take 5 to 7 days to generate an antibody response. Our first antibody is usually going to be IgM, and then we'll start to see IgG. During our secondary response, it responds more quickly. We get higher levels of antibody, especially IgG, and that helps us clear the infection better. So, we'll talk about vaccination in another set of slides. And again, as we progress through all of the different types of infections, the idea behind vaccination is that it serves as your primary challenge. So the idea would be that, let's talk about COVID, because that's what everybody talks about. Some people never got vaccinated, so they got COVID. So they didn't get an antibody response until 7 to 14 days after they were first exposed. Some people got vaccinated against COVID. So their body made antibodies to COVID within 7 to 14 days of the injection, but you didn't get COVID, right? No fever, no cough, no stuffy nose, no loss of taste and smell. But let's say you got exposed to COVID. Doesn't mean you're not going to get the infection, but it does mean that your immune response will be much more rapid, much stronger, and much more likely to clear the infection before you suffer significant consequences. So that's what vaccination does. So instead of being sick twice, you get vaccinated and only have to be sick once.